Oh, you? Yeah. Do it. Okay, so hi everyone. Thank you for joining us. We're mostly online, which is fine, but I'll do this introduction from here. So I'm very happy to introduce Bhargav. He made the trek all the way down from Florida State, and he's a mathematician as part of the math department and the biomath there. And when I met Bhargav, we were just postdocs. And I met him when I was interviewing at the University of Houston. And then after the one postdoc, he was hired by Florida State. That's pretty cool. So without further ado, please take it away.
All right, yeah. Thanks. Yeah, so yeah, first I want to thank Youngmin for inviting me and trying to get interaction between the University of Florida and Florida State math groups. I think that'll be great. It's my first time in Gainesville. Unfortunately, it's raining a lot, so I won't, I guess, walk around outside too much, but so far good vibes outside. So today I'm going to tell you about some work that I've been working on fairly recently that started maybe less than a year ago. Namely, I'm going to be thinking about delay differential equations, wherein the delay itself evolves stochastically. And then I'm going to apply it to a gene regulatory system, a very basic model of a gene regulatory system. Okay. This works. Oh, yeah. Oh, yeah. Oh, yeah. So, I'm sharing my screen. Okay.
Yeah, so you're using a pointer.
Oh, right. Okay. Yeah. Okay. So delays. I became fascinated with delays because delays are pretty much ubiquitous in biology. They pop up everywhere. Any action, the outcome of any action is going to require some time before it comes out. So there's always a delay. Sometimes you can ignore them. Sometimes you can't. But they can play very important roles and in biology. And so here I'm just going to talk about some examples and delve a little bit deeply into some examples of where delays can show up and how they can influence dynamics. Namely because not many mathematicians are not sort of so used to delays. So for example, you can model intercellular transport of signals with delay differential equations. And in particular, if you don't want to do a spatial model, these can become quite useful when you're modeling signal transport in, for example, neurons, which are these large cells with high size cell size variability. And so, for example, if you imagine a chemical signal that's produced at the soma, and then is carried down by molecular motors to the growth cone, and then activates the production of a new signal, which is carried back and inhibits the production of the original signal, there's going to be a certain delay in the time that you send the signal off and then expect the return. So there's sort of a non-trivial measurable delay in such an example. And so let me delve a little bit into what a model like this, a situation like this can lead to. So as I was describing, this mechanism I was talking about is fundamental for axon length sensing. So it's been hypothesized that kinesin motors carry a signal from the cell body to the tip, which induces the activation of a production of a new signal at the axon tip, which is carried back to the cell body by dynein motors and inhibits the production of the original signal. And so this is an example of delayed negative feedback. And if you increase the delay long enough, this is pretty typical of delayed negative feedback systems. If you increase the delay long enough, your signal will begin to oscillate. So that's what you see in this little inset here, is chemical signals are oscillating. And so there's a delay-induced Hopf bifurcation in the chemical signal dynamics, if you like. And then, more importantly, what's also typical of such examples is the frequency of oscillations. The frequency of the oscillations is correlated negatively with the delay. So the longer the delay, the shorter the period is inversely related to the delay. So the longer the delay, the shorter the. Anyway, yeah, they're inversely correlated. Yeah. Inverse with frequency. Yes. Got my words stuff. And then you can feed this into a genetic regulatory system, and you can show that an axon can reach a steady state value. And so that's just an example of how delayed negative feedback can be used to consider or study pretty sophisticated processes such as axon length sensing and even axon growth. Okay. Another fairly typical example of where delays manifest in biology are basic transcription and translation delays.
Sorry, we go back to the previous slide for a moment. So y-axis is the axon length.
Y-axis is X, Y, and whatever this chemical signal is. It's a chemical signal of at the soma. And so there's oscillations, and then in this feedback network, you have an oscillation with respect to Y, and X is a proxy for axon length. X is length, basically, and you can see that X flattens out.
Gotcha. Okay. Sorry.
So another area is just standard transcription translation. When a gene is produced, when a gene is translated, from transcription to translation, there's about a measurable 7.5 to 10 minute delay. And so oftentimes this is overlooked in modeling, but it's essential, for example, in the establishment of our circadian rhythm. So the 7.5 minute delay can help us understand and maintain and stay on track with our circadian rhythm. And that's all due to delayed negative feedback also. And a third example is hormone interactions. So if you're thinking about glucose insulin dynamics within your body, your glucose is an externally forced state variable in your body, right? You eat something and that causes some disruption to the in state of glucose in your body. And then in response to that, your body has to produce insulin and maintain sort of homeostatic levels. And so you have a delay from the time you ingest glucose to the production of insulin and the action of insulin upon glucose. And so there's a lot of influence of delays here as well. And I'll delve into this a little bit more a little bit later. But hormone interactions are also involve delays. Okay. So what are the effects of delay in biological systems? So delays can lead to really interesting dynamics. And sort of first and foremost, one very interesting example is that if you have a fixed point and you have a delayed, say, negative feedback, delays can destabilize the fixed point via a Hopf bifurcation. And so you get this nice limit cycle, which means you can draw prettier pictures in phase space. And so you get pretty orbits in phase space, all due to the existence of delay. So that's one way that delay can make interesting dynamics. So here's an interesting one in feed-forward network topologies. So if you have
before we keep going, can we? So in the one parameter bifurcation diagrams, are these? It says the parameter is signal. Is that the delay?
It's the okay. So yeah, I wasn't so precise. This is just to show things. So the signal is whatever's sort of getting the system going in these wiring diagrams. So S is just
So some, yeah, signal strength.
some signal strength. Yeah, yeah,
Okay.
signal strength. Yeah.
By the way, the flashlight is on your cell phone.
Oh my God.
Very- very best.
Thank you. But yeah, so yeah, okay, thank you. So another way is if you look at these feed-forward network topologies of, say, signal cascades or genes or your protein cascades in cells, there's a delay in the interaction of one protein with a subsequent protein. And in fact, if you read this paper by I'm just doing all sorts of okay. So that was just my home camera showing what's happening outside my house. Okay, so if you have a feed-forward network, so proteins interacting with proteins and going forward, in fact, the delays that are inherent to subsequent proteins interacting with each other actually accelerate the signaling, which is quite interesting. I don't know the full details, but you can read about it in this PLOS computational biology from paper from a decade ago. And lastly, but not leastly, delays can also induce chaos in nonlinear systems. And so this is some work I worked on in a few years ago. So glucose dynamics in your body are oscillatory, but the delay in the action of insulin upon glucose interacts non-trivially with the times in which you ingest meals. And then if you study that interaction, you can actually see that glucose becomes a chaotic sort of oscillation. You can look at the Lyapunov exponents, you get weird, strange attractors. And the invariant densities of the dynamics also change drastically. So if you have regular oscillations in your glucose state variable, you get this strange bimodal distribution in your invariant density. But whenever you have chaos, you get almost a Gaussian type of distribution. So again, this is all due to the delays. It's very interesting. So let me just delve into this a little bit more, just to show you how intricate the dynamics can be emerging from just delays. So in the glucose insulin dynamics I was talking about, if the delay in the action of your insulin upon your glucose is sufficiently fast, then your glucose, the y-axis is glucose here, and the x-axis is either time. It's a time series on the top and phase on the bottom. If the insulin acts sufficiently fast upon the glucose, then your glucose level rapidly reaches a fixed point. But as you increase the delay, you have this delay-induced top bifurcation that emerges. That's one interesting thing. And then this is just the image I showed a few slides ago. And this capital T is the time between meals. So if you force your system every 20 minutes versus every 200 minutes, you can see that you have vastly different dynamics. Namely, if you space your meals too far apart, this was an interesting finding that we found. If you space your meals too far apart, your dynamics can actually be quite chaotic. But the distribution gets more normalized. So it's kind of this, which one do you want? Do you want a more normal distribution, or do you want more regular oscillations? And so this is actually kind of an interesting idea for clinicians, because if you look at the time series, sort of the individual time series, these are the data that diabetics at home measure, right? Like they eat something and they want to be able to predict, they want to be able to predict for the future time what their sugar levels are going to be. If you have a regular oscillation, that's possible. However, if your oscillation is chaotic, then that can be very frustrating for diabetics. They eat something, they think their sugar level should be balanced 2 hours from the time they ate, but they find that it's doing all sorts of wacky things. So spacing your meals too far apart can be disruptive for a diabetic who's trying to predict their glucose in the future. However, from the clinical perspective, if you have a patient in the ICU, clinicians aren't always interested in the exact value of the measurement, but rather they're just interested in maintaining the glucose level between some two numbers. They're like, I want the glucose level to be between 100 and 130. So let's just do whatever we have to do that. And in this case, it turns out having the chaotic time series is better because your distribution is more normalized. So from a clinical perspective, having this chaos could be more beneficial. And so anyway, hopefully I've talked enough about how delays can be interesting, can induce interesting dynamics into various systems. Yeah, so now I'm going to delve into some of the more recent stuff I've done. So why are delays useful in modeling? So oftentimes, if you have some input that you're trying to that you're imparting onto a system, and you're interested in knowing what the output is relative to the input. But a lot of times you don't know what happens in between those two steps. You put something in, something comes out. What happens in between, you don't know. And if you don't know, but you sort of know how long the output takes to come out, you can just coarse grain all those mechanistic details into a delay, and that can be quite useful. And so modeling with explicit delays, i.e. with delay differential equations, can be quite useful. Okay. And why do we care about stochasticity in delays? And so oftentimes, if you're looking at, for example, subcellular processes, the subcellular environment is very noisy, lots of things going on, lots of thermodynamic fluctuations, just noise everywhere. And so you can often report a distribution of delays from an input and an output to an output. And so studying stochasticity as such can help you better understand biological or other general sort of processes. So if you know the underpinnings between the input and the output are noisy, you can try to study the system with a stochastic delay. Okay, so let's talk a little bit about delay negative feedback. So we're going to consider a very, very simple model of protein translation and auto inhibition. So you have this protein concentration, this YFT is scalar field, protein concentration. And then I is going to represent the constitutive protein production. So the gene is always on, it's just producing protein on and on. And then it has degradation or dilution, however you want to think about it. And then we have delayed negative feedback. So we have this function f, this nonlinearity here, which, and W is going to be the strength of the negative feedback. And it only manifests after some finite delay tau. And you can take f to be really any monotonically increasing function that saturates at infinity. So for concreteness, we take this Langmuir Hill function. Okay. And so what I want to do is I want to understand the stability of the fixed point of the system. So we do the linear stability analysis. Equilibrium satisfies this very simple equation, and we can linearize around that equilibrium. And then after making the ansatz, the exponential ansatz, you get a transcendental characteristic equation, which is normal for delay systems. And I'm looking for the onset of Hopf bifurcations. This is a delay negative feedback system. So I want to know when oscillations are going to manifest. So you assume your eigenvalue is strictly imaginary. And then if you plug it into the characteristic equation, out come the conditions for your Hopf bifurcation. And so a couple of things to note here is that if your delay is 0, then these conditions cannot be satisfied and your system won't oscillate, which is good, because if you turn the delay to 0, this is just a one-dimensional flow, which cannot oscillate. And so you have to have a non-zero delay to see oscillations. And so in particular, delay is crucial for the onset of periodic orbits in this system. Okay. So here's just some time series. And so if I take a delay less than what's given by the Hopf bifurcation, you see after a transient up and down phase, convergence or contraction to a fixed point. But if you take the delay longer, then your oscillations start to persist. Okay. So let's talk about stochasticity in delays. So how do we incorporate stochasticity in delays? Well, there's a number of ways. So this first way is not really stochasticity necessarily, but you can use what's called state-dependent delays, or the delay value depends on the internal state of the system. So for example, if you have in this figure here, you have stem cells that are maturing, but how long they take to mature depends on the internal state of the system. Or for example, even in genes, if your genes are on, they take 7.5 minutes to be transcribed and translated. But even when genes are off, they leak. Meaning even when genes are totally shut off, they can still produce proteins, and the delay at that point is longer. So you can have a state-dependent delay that can change or affect your dynamics. You can sample your delay from some distribution. Just a priori, you select a distribution, and every time you solve the system, or at every time point, you just sample a different delay from a prescribed distribution. Or you can use, and people like Richard Murray and have studied this quite a bit, you can prescribe a waiting time t, after which delays shift according to a stationary Markov process. So you say, okay, I'm going to be from a discrete set of delays, say tau 1, tau 2, tau 3, I'm going to spend a time t at any one of those delays, at which point I will shift to one of the other delays based on some probability distribution. And so this is just a sample time series of how your delays can change. So the way I'm gonna do it is related to this last example, but a little bit more complicated, I guess. So how do we incorporate delay stochasticity in this simple delayed negative feedback model I talked about? So I'm going to let the delays evolve according to a discrete time Markov chain on a discrete set. So you have n delays that you can choose from, say, and then the delay value in the delay differential equation is just going to jump between these values. And so you can think of the states of the delay, tau 1, tau 2, tau 3 through tau n, and then you prescribe propensities for jumps between any two states. And these fancy W's are going to represent those propensities. Okay. So stochastic switching of delays. So now I still have my very simple model, but now my tau is time dependent and evolves according to a discrete Markov process on this discrete set tau 1 through tau n. And so you can write down a master equation that describes the changes in the shifts in the state of the delay. And this is just a very generic representation, but you can write down it for more concrete examples. So if we take a 2 delay example, we know that for a fixed delay, there's a critical tau value past which the system begins to oscillate. And so let's take 2 delays, both past that critical value. You can write down the master equation, and I just scaled the propensities by this term epsilon, because I can vary epsilon now, and that's going to scale how quickly I change between different states. So let's just take a 2 delay example, and then you get what you would kind of expect is if I switch between 2 delays, both past the supercritical Hopf bifurcation point, it's a time series jumps between limit cycles that correspond to whatever delay corresponds to that state at that time. That's kind of what you expect. Okay. What happens if I start making these switching faster? If I take epsilon smaller and smaller? So something very interesting happens. So if epsilon is large, i.e. the switching is relatively slow, you do get this rich stochastic oscillatory behavior. But as I start tuning, it looks like I have a question.
If delay is a measurable but discontinuous function, can you still prove the Hopf bifurcation? If the delay is a measurable but discontinuous function, can you still prove the existence of a Hopf bifurcation? So I assume measurable here meaning as in the sense of measure theory, but so I guess when you have the delays, the discontinuous switching of the delays that you have, can you still, well, yeah, I mean, please go ahead.
So yes, I mean, there are restrictions, but measurable, discontinuous, as in the example I showed before, where I was just switching between delays. I don't know if you can prove the existence, but you can certainly do the analysis and find Hopf bifurcation points. Yeah. Numerically, yeah, numerically, yeah. Here I'll find I'll find analytic conditions for the Hopf bifurcation. But as far as a rigorous proof in the abstract sort of general sense, I don't know. Okay, so here, whenever I take the switching to be relatively slow, I get this rich stochastic behavior oscillations. But as I start speeding up the transition between the two different states, as I jump back and forth really, really fast, the oscillations contract to an equilibrium. And there's this Y effective term here that I'll talk about in a little bit. But this caught my eye when I first did the simulation. I was just playing around with simulations, and I sped up the transitions, and the oscillations were destroyed by whatever the dynamics were.
And is Q the probability density? I'm sorry, I missed that.
Yeah, it's the probability density. The probability density. I mean,
Yeah, I see. So as epsilon goes to 0, you have you reach the steady state like arbitrarily quickly.
Yep.
And so I guess for epsilon small, you end up with somehow like just a single peak in the density.
No, you don't.
Okay.
So,
Right.
so you don't. Yeah. Yeah. Good question. So you actually get a sum of things, but yeah. So, but yeah, so you contract to an equilibrium, which is okay. So the fixed point reemerges. So what's going on? So we want to understand this, and so we go to a general master equation formulation. So we go to the microscopic description of this process. So we have this probability of having m molecules of protein translated at time t, and a current delay value of tau I. And so I'm just going to write down the master equation of the simple delayed negative feedback model I had. And so I have constitutive production right here. And then I have degradation or dilution, however you want to think about it. And here's the delayed negative feedback term. And so let's just unpack this a little bit. So I have to sum over, based on the current state that I'm in, I have to sum over all possible delay states I could have been in the past. And I have to sum over all possible protein values I could have had. In particular, if I have no protein, let's say I'm in a state tau I, but tau I units ago I had no protein, then there's no delayed negative feedback I have to deal with. But any non-zero amount, I have to think about that. And so I have to sum over all possible protein terms and all possible delays. And so these terms that correspond to the delayed negative feedback are joint probability densities. So probability I have m molecules of protein at time t, current state tau I given, not given, and that I had protein capital M protein molecules tau I units ago, and I could have been in any state with respect to the delay. And the strength is still just Wi, and then I have this transition between the delays captured by the just transition Markov transition matrix, basically. And so this delayed negative feedback term, as I said, has these joint probability densities, which are kind of challenging to deal with. You can derive the equations for these 2-point correlations. But if you do that, the right-hand sides of those equations contain terms with 3-point and 4-point correlations. And you write the equations for the 3-point and 4-point correlations, they have 5 and 6 and 7-point terms. And so you get this infinite hierarchy of equations if you want to understand just the 2-point just the 2-point correlation. And so to avoid that, we can just assume that the joint probability just factors. And then if you do that,
sorry, Sarah had a comment. If you have a periodic forcing with increasing frequency, then at high frequencies you effectively average the forcing, making it basically constant. I can't tell if that was it seems like a statement. So I'm not sure what to do with it.
you have a high frequency. Sure, if you have a high frequency forcing, then instead of reacting to the peaks and the troughs of the oscillation, you probably just react to the mean in some sense.
So and that's what's happening when epsilon goes to zero, is that right? The frequency gets faster?
As epsilon goes to zero, epsilon goes to zero, yeah, the mean contracts to the
The mean, okay, so the mean, yeah, I see. The mean contracts.
So the mean does contract, the system does contract to the mean dynamics, but it's not so simple as it's switching too fast, and so you're ignoring the delays. Okay, so what we do is we assume that we factorize the joint probability. And then to do some analysis, you can't really do analysis with this discrete master equation. But to do some analysis, we invoke a Van Kampen expansion. So we assume that we can produce a large number of translated protein molecules and then scale the translated protein molecules with that number, which is determined by the physics of the system. And Taylor expand, you can get the Chapman-Kolmogorov equation for the probability density. And if you kind of squint and look at the sum integral term, this is just the expected value of the delayed variable, or the expected value of the Hill function with the delayed variable. And so you can replace that with just the expected value of the delayed negative feedback term, and you get this nice Chapman-Kolmogorov equation. So that's good. And remember, we're interested in what's happening as you take epsilon to zero, so fast switching limit. So you assume the Markov transitions are fast. And so effectively what you do, as you alluded to, you project onto the stationary measure of the Markov transition matrix, and then treat the dynamics as a perturbation away from the stationary measure. So you make this quasi-steady state or adiabatic approximation, and you can obtain this effective Fokker-Plonker-Smoluchowski equation, where your dynamics become a weighted sum of the dynamics of the individual subsystems. So this chi sub k corresponds to a delayed negative feedback term with tau k, and this phi n is just the stationary measure of the n, corresponding to the nth delay. The probability of being in the nth delay state should probably be not an n, but okay. So you just end up summing up the different. You construct a weighted sum of the different subsystems of the dynamics, and your Q is just the sum of the different probability densities corresponding to the different delay states. Okay. So let's go back to delayed negative feedback again. So again, we have this two delay example that we observe this interesting dynamics in. You can make an adiabatic approximation that I alluded to in the previous section. And if you do that, you get this nice, fairly simple Smoluchowski equation for the probability density Q. And so you're weighting the different subsystems. Here you have the delayed negative feedback with tau 1. Here you have the delayed negative feedback with tau 2. And this beta over beta plus alpha term and alpha over beta plus alpha term are just the stationary measure terms. The zero eigenvector of the Markov transition matrix. And from this, you can extract an effective equation for the mean dynamics. And so a couple of things to note here is that the effective system incorporates all the delays involved rather than an effective delay. So as I alluded to, you're not getting a single peak, but rather you're getting a sum of delta peaks. And as I alluded to before, the effective dynamics are just given by the weighted sum of the different subsystems, and the weights are given by the stationary measurement. And so you look at this again, and you can see that as you speed up the dynamics, the As you speed up the transitions between the delays, the dynamics of the full stochastic system, simulated stochastic system, converge to this effective value, effective equation, the solution of this effective equation. Okay. So why do these effective equation, like why whenever I take taus to be larger than the critical value of the critical value of the single delay system, why do I contract with an equilibrium? And so to do that, I have to do linear stability analysis on this more complicated delay differential equation now. Steady state is the same as in the non-switching case. And so you linearize and you obtain this equation, and then you make the ansatz. And here you obtain the characteristic equation for this two delay effective equation. And then you carry on as before. Excuse me. Yeah, you carry on as before, and then you substitute in a strictly imaginary eigenvalue. And here are the Hopf bifurcation conditions for this two delay case. And so just so for sanity check, you can compare it to the non-switching case. And if you set one of alpha or beta to 0, you can see that you recover the non-switching case dynamics. And so it's a completely generalized version. So here's the bifurcation structure for the two delay system. So let me describe what you're looking at here. The dotted line is denotes the critical value for the non-switching system. And so whenever you're to the right of tau 1 or when you're to the right of the dotted line or above the dotted line in the single delay system, you're oscillating. However, if you do the two delay Hopf boundary as given by these equations right here, you can see that the two delay Hopf boundary stretches into parameter space, exceeding the critical tau value, past which at which you still get contraction to an equilibrium. And so you sort of have to increase your delays to be much larger before you observe oscillations, and the oscillations will be not regular. There'll be sort of these strange, almost mixed mode type of oscillations, but I don't want to make a strict claim on that.
Sorry, Sergey has a question. Sergey, you're welcome to unmute if you'd like.
Oh, that's okay. Yeah, I just didn't want to be too interruptive. So my question about this figure is, what are the values of alpha and beta here?
What are the values of alpha and beta?
Yeah, so essentially, is it 1/2 or?
It need not.
I'm sorry, it is what?
I mean, it need not. There's really no restriction on the values alpha and beta can be.
No. But in the picture that you showed, it corresponds to some specific values of alpha and beta, right?
Yeah.
And they are.
I'll have to look at the paper to tell you.
So you're saying that the picture will not change much if you start playing with alpha and beta, right?
It won't change all that much. I mean, this Hopf boundary might shift left and right, but you'll still get this space above the supercritical Hopf point where you get contraction to an equilibrium, provided it's not 0. But okay, right. You can't take them arbitrarily small. You can't take alpha and beta arbitrarily small, but like order, let's say like 0.01 and above, you'll get a nice.
I guess if you go back, can you go back one slide? So if alpha is equal to beta,
yep,
right? So you have the sum of sines divided by the sum of cosines. So you can extract the frequencies from that, right? And one of them will cancel, which is the shorter one, and the other one will probably remain, which is the longer one, right? So you'll get frequencies like tau 1 plus tau 2 versus tau 1 minus tau 2.
If alpha is equal to which equation are you looking at? I'm sorry.
I'm looking at the equation in the right box on the bottom, where you have a linear combination of sines in the numerator and cosines in the denominator. So if alpha and beta are equal, they cancel out, right? And then you have the sum of sines divided by
Oh,
the
yeah.
sum of cosines, from
Yeah,
which,
yeah, right, yeah.
yeah, you can use the, trig identities to extract
Sure.
the differences in the sums.
Sure.
Right. And so I guess one of them will cancel out, right?
Yeah, because
Effectively, you will go from tau 1 and tau 2 as two independent delays to just their sum.
you get their sum, but why would the other term cancel out? Why would the difference cancel out?
So the sum of the sines versus the sum of cosines can be expressed as products, right?
Sure.
And then one of the terms in those products will cancel out on top and the bottom.
Oh, I see what you're saying. Okay, yeah.
So I'm guessing that's why you're seeing this increase in the delay, because effectively they start acting together, like tau 1 and tau 2.
Mm-hmm.
Just add it up. No?
I'm sorry, can you say that again?
So tau 1 and tau 2 will be, tau 1 plus tau 2 will be the effective delay.
Ineffective delay in this case that you described. Yeah, that's right.
Okay. Well, anyway, thank you.
Yeah, sure. In this case that you described, absolutely. Anyway, okay, yeah. So you get this region in above supercritical state, the tau for the one delay case that you won't get oscillations, and then here's just another view of that. So here's the bifurcation structure with respect to the switching rate, which has not been looked at in the literature as far as I know for delayed systems. So as you slow down the transition between the different states between two taus, the black dots here correspond to the fixed point or the max and the min of the oscillation that's been observed. And the blue lines are the max and min of the oscillation for the state with tau 1, and the red are the max and min for the oscillation with tau 2. And so you can get a Hopf bifurcation as you vary epsilon as well. And then on the right-hand side, it's just another view of that. So you're looking at the peak of the power spectrum. Okay, anyway, so we derived an effective equation, assuming the fast switching limit between delays. So what's interesting is the equation that we came up with for the effective equation, where we have a sum of subsystems, has been used sort of as without justification in other papers, and so we, I guess, gave that justification to provide motivation for that equation. And this time scale of stochasticity has a strong on the system dynamics, and there's a lot of emerging literature now on switching systems and how stochastic hybrid systems or piecewise deterministic Markov processes can exhibit vastly different behavior compared to the non-switching cases or noiseless systems. And so we've kind of added to that literature with this paper. So if this paper got your juices flowing, here's the citation for it. It was published this year in Royal Society Interface. And I'd like to acknowledge my collaborator, Chris Miles, who's an assistant professor at UC Irvine. Did a lot of work in helping visualize the different things. Okay, yeah, thanks.
Are there any other questions for the speaker?
I can always ask an extra question if you.
Excellent.
So can I ask you about the theoretical foundations for these calculations? Because if you consider a system with fixed delays, right, it has regular solutions. I mean, it's just a regular ODE with differentiable functions as solutions, right? But if you start jumping between discrete values of the delay, as you do in your Markov chain setup, right? Then the solution, the right-hand side becomes discontinuous. effectively. And first of all, how do you even guarantee classical solution? Or if you're dealing with non-classical solutions, then how do you justify all of this linearization, et cetera, doing all these characteristic equations and stuff? Right?
So the stochastic system has a discontinuous right-hand side, but the effective equation does not. So I think you can proceed as usual with linear stability analysis for the effective equation.
Okay.
And then as far as the master equation and discontinuous right-hand side.
But with the stochastic system, why does it even have a solution?
I mean,
typically you assume that there's some stochasticity, right? It's still a continuous function, like the white noise and stuff.
Yeah. I mean, this is a fairly common thing to. I mean, not common. It's kind of an emerging.
No, I understand that you can write the code, plug it in, simulate, and so on and so forth. But is there an underlying solution behind all of this? Like what is the functional space where it lives, et cetera, et cetera?
Oh, I mean, I don't know.
Okay.
Yeah.
Okay.
Yeah.
Thank you.
Yeah. Thanks.
Anyone else? All right, well, thank you so much, everyone. Really appreciate your time. Let's thank our speaker again. And if you want to join us for lunch, just let me know. And I'll see you all around, and he'll meet you in various meetings. Okay, take care.
Thank you. Hey, Dan, how are you doing?